So our next speaker is Alexandre Renaux. Oh, all right, from the Université Libre de Bruxelles.
Yep.
I feel like I'm damaging the language
No,
every
it's
time I speak.
completely fine. Okay, good morning everyone. Thank you for inviting me. I'm Alexandre Renaux. I'm a PhD candidate at the Inter-University Institute of Bioinformatics in Brussels, between the ULB and the VUB in Belgium. And today I'm gonna introduce in this talk what are oligogenic disease briefly, and how ORVAL, which is a predictive and explorative platform, can help researchers in discovering potential pathogenic variant combinations. So in Europe, it's estimated that around 30 million people are affected by a rare disease. And it's suspected that a majority, around 80%, could be of genetic origin. As you know, it's often difficult to associate the phenotype of a patient with genetic causes. This is because of the availability of only a small number of cases for this kind of disease, cases or families. And also because of some phenomena like reduced penetrance, genetic heterogeneity, and phenotype variability. So those difficulties are often referred as the missing heritability problem. And as you can see in this chart, the genetic diseases are not only divided between the monogenic diseases and the polygenic disease. It's actually a continuum, where on one side you have the monogenic diseases explained by rare variants with high penetrance and happening in a single gene. And on the other side of the spectrum, you have the polygenic diseases that are formed with an accumulation of common variants with low penetrance and happening on multiple genes. And in the middle of this continuum, you have the disease with intermediate penetrance and frequency. There is an emerging hypothesis that those diseases that we refer as oligogenic diseases could explain for some disease that's missing heritability. That was for us a strong motivation to do this research. And so we wanted to go step by step to a higher level of complexity. And so for that, we started with the simplest form of oligogenic diseases, which are the digenic diseases. So you have multiple cases from clinical papers about digenic diseases, for example, on the Hirschsprung disease, on some case of congenital deafness, or the Bardet-Biedl syndrome, for example, and some cardiopathies also. So in our team, we made a database called DIDA, for Digenic Disease Database, that collects around 250 combinations causative of 54 diseases. This database, when it was published, received quite a lot of attention from the geneticists because it was the first database of this kind. So in the digenic diseases actually are defined as the case where when you have mutated alleles at two loci, it explains better the phenotype of the patient. And so you have three types of models for the digenic disease. The true digenic, where you have a perfect synergy between the two loci, and so you need to have the two mutated alleles to develop the disease. Then you have the monogenic plus modifier model, where you have one gene, what is called often the driver, and then a modifier. And so if you have the driver, you might have some phenotype response, and the modifier will change the severity or the age of onset. And then you have the dual molecular diagnosis, where you have two Mendelian variants at two different locations, and those will give you blended phenotypes, because sometimes diseases are just a combination of different phenotypes. So in our team, we developed machine learning methods. I won't go into a lot of details for this one. That is able to predict the different types of digenic diseases. So it's based on random forests, and it's learning from different features describing the variant level, the gene level, and also the combination level with if there is a relation with a pathway. So you can have more information. It has been published, and I'm happy also to discuss about it later. So the next step was to predict if a variant combination could be potentially disease-causing or not. And so for that, we use the DIDA, so the database I presented earlier, in combination with 1,000 genome as a control data set. So this method, called VarCoPP, for variant pathogenicity predictor, is generating basically. It's an ensemble method, so it's generating several classifiers, 500 exactly, that are sampling 1 kGP as a control and DIDA. So it makes different samples and create 500 classifiers, and the results of this predictor is a majority vote amongst all the classifiers to tell you if the combination is potentially disease-causing or neutral. So where we don't know actually. And so these results, this class as disease-causing and neutral, is supported by two scores, the support score and the classification score, giving you the information on the support amongst all the classifiers and whether those classifiers are predicting the disease-causing class with a lot of accuracy. And so this method, this machine learning method, is based on selected predictive features that have been selected quite carefully to avoid overfitting, and it's also describing the combination at the variance, gene, and combination level. So for the combination this time, it's using the biological distance, which is a measure of how far two gene products, so the proteins, are in protein-protein interaction network. So this method has been published written very recently, actually, and so it's available here, and you also have the code available on the GitHub if you want to reproduce the performance tests. So the next logical step for us, and it's kind of a big leap, was to go to oligogenic diseases. So the traditional approach for some people who tried that is to identify variant with monogenic predictors. So you predict the nodes with classical predictors, and then you try to use network data to link those different predicted nodes. Using, for example, PPI co-expression. The problem with those approaches is that it cannot really identify synergies because it's based on it's univariant methods, and also because those predictors are designed for severe effects. The other problem is that, as you know, networks, biological networks, you can have some links that are unknown, so it can be also another problem. So in our approach, we are using actually VarCoPP, the predictor I presented earlier, to have predictions that are bilocus. So bilocus is basically for us a synonym for digenic. And we predict edges, actually, not nodes. And so we predict potentially pathogenic relations between two genes. And so from those predictions, those digenic more bilocus predictions, we can aggregate the information then at the gene level to create a network, and then after it annotate it with other network known biological information. So that led to the creation of a platform for researchers to analyze their variants and to discover potential variant combination. So just a side note for the name. So it's coming from an abbey, a monastery in Belgium called Abbey d'Orval. I really recommend to visit it if you go to Belgium. It's beautiful. And the actual reason why we chose that name is that the monks there are brewing an amazing beer called ORVAL. So yeah, back to the platform. So in this platform, you can submit variants in a VCF or manually if you have like a couple of variants you want to test. And you can then filter those variants based on a gene panel, based on mineral allele frequency and other kind of criteria. And then it handles submissions with a new unique ID, and then you can retrieve your results for 7 days. And then this platform embeds the 2 predictive algorithms I was talking about before. And on top of that, also this kind of network approach I was mentioning with contextualization to annotate them with PPI, serial allocation, and pathway information. So when you go to this platform and you submit your variant, it will make combinations, and then it will predict the combinations as being either predicted neutral or predicted pathogenic. And so you can access all the details on the table on the right with the scores. And then these predicted pathogenic are divided into 3 different zones. And in, for example, the 99% zone, you can expect to have 1% false positives. So this is based on control data sets from 1KGP, where we made basically neutral combinations, and we try to assess the number of false positive. Then when you select a specific variant combination, you can have access to an explanation on why the predictor took that decision based on feature contribution. So it basically decompose the decision and you try to know then what are the features of the machine learning methods that contribute the most to the disease-causing or neutral class. And then you can also analyze, using the Digenic effect predictor, the type of model, Digenic model, that is predicted. This is also another result that you get on the overview page. And the goal is to kind of represent in a network way what are the kind of patient network, pathogenic network in a way. And so you can see here that this network can be explored. You can click on a specific node and then get the information, and you can also filter based on some criteria like the scores, and then you can click on a specific module to explore it with protein-protein interaction. Like the network. So in the center you have your selected gene sets, and outside are the first level interactors interacting with this set. And then you can also have access to the probabilities of being in a certain cellular compartment and highlight it on the PPI. You have also contextualization on the pathways, so it's using Reactome to enrich your gene sets with some pathway information and visualize it. So that's the platform, and so we tried a recent case that was published in 2019 of like more complex, so Digenic, to see if that could work on ORVAL. And so this is a patient suffering from mild hypertrophic cardiomyopathy, and they suspect there is a Digenic relationship between 3 genes, and so they give access to the variants, obviously. And so we tried those variants on the platform, and ORVAL confirmed the suspicion of oligogenicity with these 3 genes and variants. And so if we go into more detail, we can see that there is a clear relation from known network and pathway information between 2 genes, so the DSC2 and DSG2, but another one is not part of it, so it could be like some kind of blended phenotype. And then we also tried the asymptomatic son, so healthy, that doesn't have a mutation on one of the genes. And we could see that for the combination left, it was predicted as neutral, which further supported. So as a conclusion, ORVAL is a web platform that integrates different resources to predict potentially disease-causing oligogenic variant combination, to explore the prediction as a network, and to understand the results with some interpretations and contextual informations. Our plan for the future is to validate the predictive algorithm and the pathogenic network on multiple cohorts, to provide also for geneticists who are really interested into that, trio analysis where you can submit the parents also, to use the phenotypic data, at the moment it's not there yet, also to improve the data database, to add new data and also make some kind of community curation, and to improve the predictor VarCoPP with new features and disease-specific predictors. So this platform is available online at this website, orval.ibsquare.be. It has been published very recently on NAR special edition. And yeah, I would like to thank you for your attention. Thanks to my supervisor team and collaborators and the funding agencies. And if you have more questions, if you are interested by this work, I will present a poster at U10. So yeah, and I'm happy to take some questions.
Can you share that website again?
Yep, sure. Yep.
So questions? Yeah?
Thanks, nice talk. I was wondering how often it happens that the predicted effect is not merely explained by the additive effect of the pathogenicity of each of the variants, which brings us to how your work relates to previous work on epistasis, or eventually whether you have tried to cross this with data from synthetic littles.
Yeah. So by pathogenicity predictors, you mean like the CAD scores and these kind of things? Yeah, so usually if you look at the contributing features, so I have like an additional slide on this. Yeah, you can see that it's not only the CAD predictors, like the individual scores for the different alleles. So they contribute the most, obviously, but then it's a synergy of all the different features that will give you different predictions. So you have also for example, the distance in the PPI. That is also something important. And considering epistasis, so that's something I want to explore for my PhD specifically. So this was like kind of like a project, but I want to go further into this. Yes, I want to explain it in a more like fundamental aspect. Yeah.
So, any more questions? I have one. Mm-hmm. So it's really cool to try to figure out the pathway interactions between genes that have been damaged. I'm kind of curious. I suspect that the mutations that you're looking at are, if you have these 3 genes that you can identify, let's say, for argument's sake, they would probably affect the binding of these genes. So it's probably not a pathway in a just a general sense of the term, but actual physical interaction. Have you actually considered this, what the story there is?
So for the physical interactions, yeah, we are investigating, so the PPI, the general PPI, but it doesn't mean sometimes PPI is an aggregation of physical interaction, activation, and different kind of interactions. So we want to go into more details now to know if what's going on in terms of if the two, for example, you have different cases. There is a nice paper actually about that, describing the epistasis effect. and so you can see sometimes it's involved in a complex, so you have a real physical interaction. Sometimes it can be very distant. So yeah, I want to explore that now, but it's at the moment ORVAL doesn't provide this kind of detail. It's not as fine-grained as that.
So I'm just sort of suggesting if
Yeah,
that's
yeah,
your
yeah.
interest,
It's a good idea.
it would be really curious to see
Yep.
if the interacting genes have mutations that affect the binding sites. So,
Yep.
but what you're basically doing is
Exactly.
damaging.
Yep.
Thank you. That was
Thank you very
excellent.
much. Yeah.
One more.
I don't know. Yeah.
Ah.
What? Right now.
Yes, there's a combinatorial number of interactions. So when you apply this in practice on a genome, what is the precision of the method?
So yeah, that's a very interesting question. So at the moment, it's not very advised to do, so even exome. So it's recommended to go at the exome, not the genome level, because the training data set was trained on kind of like coding sequence, etc. So it's recommended to upload exomes, but on top of that, we also upload a gene panel. We have on the paper of the predictor VarCoPP, there is an analysis of that, and they recommend to upload a gene panel of 100 up to 150 potential genes to have a good precision and recall. Yeah.
So let's thank the speaker one more time. Our next speaker is from the Technical University